well, welcome back to the Classics of Immunology Journal Club. And before I forget, because I do that now and then, don't forget to like the video on the YouTube channel. Click on the bell to get notified for more. Subscribe. Check out my website by scrolling down and getting to the kendalasmith.com website, and then click on the writing tab, and you'll find all the classic papers that I've picked out as what are classics, including the one we're going to discuss today. Today, we're up in real time, as I've said in the last couple of weeks. We are going to review a paper that was published in Nature on January 2nd of 2020. What a formidable date that is, because it was right before the pandemic hit, and within the next month or so. So this paper is by Patricia Darrah, 21 other authors, and then Robert Cedar from the Vaccine Research Center at the NIH. And it's all about tuberculosis vaccines. And this paper, in my way of thinking, is probably it's really a deal breaker for the vaccine field. So we see if you agree with me. It's entitled Prevention of Tuberculosis in Macaques After IV BCG Immunization. The background on this paper is quite interesting because it takes us back to the beginning of immunology in 1798 with Edward Jenner and the smallpox vaccine, because basically what Jenner did was that he took what he felt was going to be an innocuous bug, the smallpox or the pox virus or the pox exudate from milkmaids' hands or the udders of cows, and inoculated humans, individuals that had already had smallpox and survived. First he did that, and then he did people who had not had smallpox. And then he went even one step further because he challenged these individuals with virulent smallpox material. And he was able to show that the cowpox immunization, vaccination, and that's where the word vaccination comes from, because in Latin and in French, vache means cow. And the vaccine, which was live virus, because they didn't know what a virus was at that time, but they used that word in the publication. And virus basically has, if also from Latin, as I recall, means poison. That was an attenuated live vaccine, and the first vaccine ever created, essentially. Nothing happened for almost 100 years in the vaccine field until Louis Pasteur came along and popularized vaccination, both as a prophylaxis against infectious, contagious diseases, but also as a therapeutic once somebody had become infected, for rabies, for example. And Pasteur was the one that really popularized vaccination and made it something in the end of the 19th century and into the early 20th century then. And Pasteur, the whole theory about how the immune system works is that you had to, for a vaccine to work well, it had to be a live, attenuated microorganism, so that he spent several years trying to attenuate virulent microbes in his laboratory by different culture methods, and he came up with a couple of different ways of going about it. But he was adamant that it had to be a live vaccine. The bug had to be alive. And the reason being is that his theory about how the immune system works, or how immunity worked, was that if you put that live bug into people, that bug would deplete the host of vital essential nutrients. And so then the host would be changed, and it wouldn't be able to support that particular bug anymore, because they wouldn't have the trace nutrients. Now, of course, we now know that wasn't true. That was totally false. But from a microbiologist standpoint, you can see why they would think that way, because that's what happened in vitro, in broth or else on petri dishes and so forth. If they didn't have the right ingredients in their culture media, the bugs wouldn't grow. And so it seemed logical to Louis at the time. So the next thing that happened in the early part of the 20th century when Robert Koch from Berlin, from Germany, who was one of Pasteur's big rivals. They hated each other. And Koch wanted to develop a vaccine against tuberculosis. That was his quest. And he tried various methods and so forth, and ultimately came up with what became known as purified protein derivative, or PPD, and which, when inoculated into the skin, would form a delayed type hypersensitivity reaction. Now that particular method and so forth and attempt at vaccination didn't work for tuberculosis, but what it did evolve into was a test for people to find out whether or not they'd been exposed to and had developed immunity, if they didn't have any overt symptoms, to the disease. And so we use that today, essentially in the clinic, as a test for prior infestation and infection with tuberculosis. Now, the next thing that happened is that in 1924, at the Pasteur Institute, both Kamath and Guerin were investigators at the institute, who took one leaf out of Edward Jenner's playbook, and they took Mycobacterium bovis, a mycobacteria that had been isolated from cows, and they developed a live tuberculosis vaccine by using Mycobacterium bovis. And that vaccine is now used in many countries around the world, particularly in Europe, but is not used in the United States because the clinical trials never were all that convincing that it was helpful in preventing infection with tuberculosis in adults. It seemed to work really well in newborns in protecting them against getting a very serious lethal infection. And so that's where things stood with BCG, Bacillus calmette guerin. It wasn't until then, after the next decade, in 1937, a yellow fever vaccine was developed out of a live virus, and yellow fever is basically a hepatitis. It's a liver infection. An investigator was successful in serial passaging of the virus in mice, after another, which was one of Pasteur's tricks to try to attenuate these bugs. But then it rested, and everybody, believed Pasteur. It had to be a live bug, and if you couldn't attenuate the bug, you couldn't make a vaccine. And of course, throughout the first half of the 20th century, bacteriology was really a big thing because people were isolating all these bugs and so forth. But it wasn't so easy to attenuate these bugs, as people found. And so Pasteur's dream of preventing most infectious or contagious diseases and then treating them also with therapeutic vaccines never came to pass. It wasn't until World War II when they needed a vaccine against influenza virus. Because recall the influenza pandemic that happened in 1918 during World War I. It killed almost 50 million people worldwide. That was huge. There were only 2 billion people in the whole world at that time, rather than 7 or 8 billion that we've got now, or 9 billion. I can't remember. But because they were so worried that their recruits were going to develop, they're going to put them in the barracks, get them all crammed together, they get the flu, and they all die before they got to be shot at the front. They started clinical trials in the recruits using formaldehyde-killed, fixed influenza virus isolates. And of course, at that time, they were able to measure the virus by the hemagglutination inhibition assay that was developed primarily by Sir Macfarlane Burnet down in Australia. And so they developed this vaccine. It was a killed vaccine. It was really the first one. And they basically took it from clinical trials directly into all the recruits in the United States. And it worked. One of the investigators, turns out, on that trial, on those trials with the recruits, was Jonas Salk. As a young lieutenant in the Army at the time, he was one of the clinical investigators running those trials. So he knew that Pasteur's whole dogma that you had to have a live vaccine was wrong. And so the next thing that happened was when the war was over and Salk went home, and he started to develop a polio vaccine. And the Salk polio vaccine, he did clinical trials in 1954. In 1955, it was declared that it worked. And the rest, as they say, is history. And it was all because Salk really pressed forward with making a killed vaccine. It wasn't until, 10, 15 years later in the 60s that people started trying to make live attenuated vaccines for all the childhood viral infections, mumps, measles, rubella, and so forth. And those worked. And to this day, that's what we use for our MMR vaccine that we use for individuals, children. Now, and so that's the background. And BCG as a live attenuated vaccine for tuberculosis never really was developed. And so the question is, what was wrong with this vaccine, this Mycobacterium bovis? Well, Robert Cedar and his group at the VRC in Bethesda decided to look into this more carefully. And in the introduction, they go through their reasoning for these experiments. First of all, because of way back from Jenner on, the vaccination for most of these microbes was given into the skin or underneath the skin, intradermally or subcutaneously. Given that way, it had variable results in clinical trials from 1924 on as to whether or not it really worked or not, and if so, how well it worked, and that sort of thing. I mean, you have to remember that clinical trials back in the 20s, 30s, 40s, and so forth were not as sophisticated and so forth in how they did these kinds of trials, which probably had a lot to do with what the outcomes were. But had been decided by then, anyway, by now, that T-cell immunity was really required to control tuberculosis and to prevent clinical disease. The T-cells had to be activated and recognized. And so just like in smallpox vaccination, you had to have a strong DTH response, a strong cell-mediated immune response. And the thing is that the route of infection in tuberculosis is through the nasopharynx and down into the lower airways, directly into the lungs. And intradermal or subcutaneous vaccine administrations don't really, result in induce high frequencies of resident memory T cells in the lungs, which is where the infection occurs. And so there were experiments in the 1970s that they reference in the paper in the introduction, where the people had done some experiments with aerosol immunization with BCG. And they also had done experiments with IV, intravenous administration of the vaccine, which sounds pretty drastic if you think about it. But when you inject the vaccine into the venous system, ultimately it gets dumped into the superior vena cava and right directly into the right heart. And what does the right heart do with all that stuff? It pumps them right directly into this huge vasculature into the lungs. So that's basically the bottom line to this paper. That works. And so they started with the hypothesis that if you gave them sufficiently high doses intravenously of BCG, then you could generate large numbers of T cells in the lungs of individuals. And they set their experiments up in the materials and methods of this Nature paper. They figured out their sample size statistically and so forth, that they need to have at least 10 individuals, subjects per group of the different things that they were going to test. They did these experiments in macaques. For their vaccinations, they used a standard strain of BCG that is obtainable from the Statens Serum Institute in Copenhagen, and they used a standard strain of called Danish number 1331. They had different groups of macaques that got different doses and routes of this vaccine. They got an intradermal low, which was sort of consistent with the traditional way that you would give BCG, of 5 times 10 to the 5th colony-forming units per injection. Now, 5 times, so that's 500,000 CFUs. That was the ID low, the intradermal low dose. Then they decided to bump it up to an intradermal high dose of 5 times 10 to the 7th CFUs. So that's 50 million units rather than 500,000 units. Then they compared that intradermal high dose with intravenous at the same dose, 5 times 10 to the 7th CFUs. And they compared that then to aerosols through a nebulizer, so that you breathe into the lungs at the same dose, 5 times 10 to the 7th CFUs. Then various immunological tests were done that are pretty routine nowadays, which is very, very good, because 40 years ago, we didn't have any of the tests that these people used in these experiments that we're going to talk about today. In order to find out whether or not these animals were really protected against lethal tuberculosis, they challenged them by instilling via a bronchoscope 6 to 10 months after the vaccination. They only had to give them anywhere from 4 to 36 colony-forming units, just a few bugs, and that would be a lethal dose of tuberculosis into the lungs. You can see why that was a pretty contagious disease. In the results, and now we'll go to screen share, and I'll show you figure 1. This figure in the A portion, we have bronchoalveolar lavage data, cells from the bronchoalveolar lavage. And what we've got here on the ordinate here are the log to the base 10 cells per bowel for each collection. And so right up here in the upper left is the total cell number. And the key to these different lines is over here on the right. So we have intradermal low, intradermal high, intravenous, and that is in red. And then aerosol, and then a combination of aerosol and intradermal. And you can see that the total cell count there's a 5 to 10-fold increase in total cells that are almost all T cells. This is T cell count, CD4, CD8 positive T cells, gamma delta, and then mucosal associated T cells. And here is INK macrophages. Natural killer cells go up, they blip and come back down, as do B cells, neutrophils, and so forth. So intravenous administration of the vaccine, and this results in a marked increase in the bronchoalveolar lavage cell population. Then if you don't look down at the B portion of this figure, we have the same immunization groups. We have ID low, ID high, intravenous, aerosol, and a combination of aerosol and ID. And in this portion here, these are CD4 positive cells that activated in vitro from the peripheral blood cells in these animals. And then they've measured intracellular cytokines for interferon gamma, IL-2, TNF, and IL-17. And so the striking thing is here, both in the intradermal high dose and in the intravenous dose, we've got a lot of positive cells going on in here. Whereas the ID low, which is the standard BCG dosage for the vaccine, or even in aerosol or the combo, there's nothing going on. And in the C portion, these are CD4 positive cells in B and C. These are CD8 positive cells, and you can see it's the same phenomenon. And the IV administration of the vaccine is what really seems to be working. Then in the D portion, we have, so B and C were the PBMCs. Same kinds of things were done, activating the cells in vitro with PPD, but now these are from the bronchial alveolar lavage. And you can see that there's in the IV dosing, there's a huge increase in cells that are capable of making the cytokines. And so these are CD4 positive cells. This is percentage, and this is cell number down here. Same thing with the IV, looking really good. And these are CD8 positive cells, and the same thing with the IV, looking very good. And then they looked at different genes that have been associated with protection against tuberculosis, and I'm not going to go into all those data right now, but in the second figure in this paper, they've now taken those animals that had really good responses to the vaccine, the intravenous vaccine. Now they've challenged these animals with virulent TB, and they followed the infection in these animals. And this is the challenge, was done 6 months after the vaccination, and they only had to use 10 CFUs of the really infectious MTB strain, Mycobacteria tuberculosis. Then they looked at the endpoint 12 weeks later after the challenge, and the infection was tracked with fluorodeoxyglucose, PET CT imaging, and that's what you see here in the lungs of these animals is the PET CT imaging. And then this is over here in the A portion is the log increase in the FDG. So here are what you see here in the this is unvaccinated, ID low, ID high, intravenous, aerosol, and the combo. You can see that the unvaccinated, the yellow and red are the increased in cellular metabolism because there's all kinds of infection going on in these animals. And these are the weeks after the MTB challenge, 4, 8, 12 weeks in each of these instances. So here you can see the intradermal vaccination really didn't protect them very well. The higher the dose, you're starting to see a better response. But here, look at IV. There's basically only one animal that had a positive result. All the rest of them seemed like they were protected in this instance. The aerosol was no better, and the combo of intradermal and aerosol was no better. If you look down to the lower half of this screen, what you have here are the lung FDG data, and you can see that almost all of everyone except the intravenous vaccination, they had a lot of infection going on. And the same thing here, if you're just looking at the CFUs of the actual bacteria in these animals, really the intravenous vaccination was the only one that really worked. So I'm going to stop screen sharing at this point in time. The there's two other figures in this paper, figure 3 and figure 4, and I really urge you to read this paper carefully, because if this isn't a classic, I'll be a monkey's uncle, you know. Of course, we have to wait to find out if you're good to be in the classic car industry. You got to wait 25 years to figure out whether you got a classic or not. The bottom line of all of the experiments that were published in this particular paper is that the route of the BCG vaccination is the primary determinant, and for control of mycobacteria tuberculosis. And the intravenous route is the only thing that really worked. In the discussion of this paper, they make several points in the discussion. The first is this study, they say, is a major step forward in TB vaccine research. And I venture to say it's a major step forward for all vaccines, because the important takeaway from this paper is that the route, the portal of entry of the bug into the system should be the same portal of entry of the vaccine. We talked about that a couple of weeks ago in another study where we were looking at vaccination through the nose. And they speculate that there could be at least 3 different immune mechanisms responsible for their really good results. One is the high T cell response in the lung tissue and its correlation with a marked decrease in the amount of virulent TB bugs in the lungs. The second thing they say is, and this sort of fundamental immunology, could be due to some antibodies might be involved. And also there had been some reports about so-called trained immunity in macrophages, and they discuss that and say that that couldn't have been going on there in this particular series of experiments. They then go on to say that there are several unique immune responses that they detected from all of their tests and all the things that they could do in order to find out what happened as a consequence of their vaccines. As they said, first of all, there were really a huge number of MTB responsive T cells in the BAL and in the PBMCs with IV administration. And in data that I didn't show you, they show that there is a unique transcriptional program profile in the BAL cells. And also they found large numbers of T cells in the lobes in the lung tissue of every lobe of the lungs of these animals for months thereafter, after the vaccine was administered. So they conclude that, and this is what they state, I quote, this is a paradigm shift in vaccine development to prevent latency, active disease, and transmission, and not solely on preventing infection. That's important. That's basically what all vaccines hopefully will do, is that they'll prevent overt disease. And in the COVID vaccine era that we're in now, for the first time in the vaccine world, we've had vaccines that prevented infection. But now we're in a stage where the virus has outsmarted the immune system and is mutating around those very spike protein receptor binding domain vaccines. And we're having to rely on our T cell mediated immunity to prevent us from having serious disease, hospitalization, and death. And they go on to say then in the last part of their discussion that the IV route may improve the protective capacity of other vaccines. And here you would read COVID. And they go on to say that this study represents a benchmark for future vaccines. And I agree with all of their statements. And to be able to state these kinds of things in their discussion and in their paper, well, that's the kind of thing you would hope for your whole career in science, because this is really a breakthrough. And this is a paradigm shift. And this should be taken seriously by all people interested in immunity and in vaccination to prevent infection or serious disease. I'm sorry. So I'll stop there. Don't forget to like the video if you did, and click on the bell and subscribe and check out the website. Read the paper. That's the most important thing out of this whole business. And come back next week. when we'll discuss another real-time paper in immunology. Thanks. If you're new to immunology, or you just want to delve into a certain aspect of this large field, and you want to get a grasp on things, you should buy my new book, Molecular Immunity, because it's a chronology of all of the important experiments and discoveries that have been made in the last 60 years. It's concise, but it's also very comprehensive. And it's the only such book that's been written since 1970. So you can buy it on Amazon, and together with the YouTube videos of the Classics in Immunology Journal Club, you can't go wrong.